Thank you, everyone, for attending this joint webinar about using animal-free VitroGel Hydrogel in comparison to animal-based extracellular matrix for Xenograft PDX and CDX models. It's between TheWell Bioscience and HC Bioscience, the Wuxi Apptec company. My name is Christopher Oak, and I will be the moderator for the session. There will be some polls and session materials available during the webinar, and you're welcome to ask questions during the webinar, and we will try to answer them afterwards when it's finished. There will be two parts. So if John, you can actually go to the next slide, please. There will be two parts of the webinar, two sessions. There'll be a quick introduction of the speakers, a little bit about TheWell Bioscience, a quick overview of the VitroGel Hydrogel and the Xenograft PDX and CX applications, data and results. And then HC Bioscience, the Wuxi Apptec company, will go over their company application and data, comparing the animal-based ECM versus with our VitroGel Hydrogel. And they will conclude with live question and answer in the audience. Okay. Next, John. I want to introduce the speaker. So John Wong would be to do the first part of this session. He is the founder and CEO of TheWell Bioscience. And the next session would be with Minqi Wang. He's a scientist too with HC Bioscience, again with the Wuxi Apptec company. And from there, I will turn over to John to start the webinar.
Sure. Thank you, Chris. Thank you for your introduction, and welcome everyone. Okay. So the first section of this webinar, we were talking about the TheWell Biosciences hydrogel system, the xeno-free hydrogel system. And I want to give you a quick introduction about TheWell Bioscience. We are focused on 3D cell culture. We are pioneering the 3D biomimicking platform to drive the advanced biomedicine and improve the personalized medicine for every patient. So we provide a platforms of the synthetic hydrogel, but we also do that three focus area. One is the precision medicine. We use our platform to generate the 3D disease model or generate the 3D tissue model for the precision medicine, for the drug screening applications as well. And we also focus on the cell therapy. The way we contribute for the cell therapy is that our system can be used as a delivery systems, injectable delivery system. So we can add the cell like a mesenchymal stem cell and use them to deliver to the in vivo model. We also use our platform to culture and maintain some stem cells and use these cells, harvest them out of our hydrogel system and use these cells for the cell therapy application. And the third areas we focus on is the biomanufacturing. We realize there's a bottleneck issue so when people want to scale up the cells in the traditional way, including the microcarriers or like traditional 2D cell culture systems. So using our hydrogel-based 3D cell culture system, we are able to easily to scale up the stem cell, mesenchymal stem cell, and some other adherent cells in our hydrogel system. And with this system, we can use this to support the cell scale up, use it to support like proteins or vaccine production, or even use it for the exome production. So those are the three areas we focus on. So right now, the TheWell Bioscience will provide over 28 different products. And our product has been used more than 500 different research labs, the institute, biotech companies over the world. We have over 100 different publications, and the product has been used for more than 250 cell types. The idea for this next generation of the hydrogel system is that we all know that animal-based materials has a lot of these unstable, inconsistent compounds. And instead of like taking everything from animal and has a lot of these undefined compounds, our idea is that to build a system from bottom up. And so we are able to control the property of the hydrogel system, including the mechanical properties, including the biological functions of this hydrogel system, and also degradability. And with this well-defined system, we are able to add the growth factors, the supplements, whatever we want to make the whole system is a well-defined system for 3D cell culture and the advanced applications downstreams. And because of these synthetic properties, we can also manipulate the system, and then also to make the system fulfill some industry standards. We make it a xeno-free biological function. We can also make it a room temperature stable and has the batch-to-batch consistency. Also, we will consider the downstream application, have the easy cell harvesting, and make it as the injectable hydrogel for the in vivo application. With that, the system can do the many different type of the application, including some 3D cell cultures, some like a 2D coating cultures, co-culture. We can support some static suspension cultures for the cell scale up. And we can also use it as the hydrogel cells a bit, and then use it to scale up some adherent cells. Of course, that we can use the system for the animal injection as well. And with these all the different yielding methods, we can create like a 3D spheral, 3D cell networking structures, organoids, co-culture system, and angiogenesis assay. We can also do a lot of different invasion assays through this functional hydrogel, and also use it for the cell scale up. But today we're going to focus on this in vivo application for the xenograft application with this hydrogel system. So the hydrogels we create has the unique shear thinning and rapid recovery properties. When I say that, is that when the hydrogel form, it can maintain its mechanical strength. However, when we use that some shearing force, like you use the syringe to inject, actually you're shearing the gel and you reduce the mechanical strength, the gel can start to flow like liquid. And once these mechanical strengths stop, the gel can recover and become the hydrogel again. With these unique injectable properties, actually is the make the gel is the extra material for the animal injection. With that, the actual sample preparation is very simple. We make the gel as the room temperature stable hydrogel. So as you can see here, the gel is in a liquid form at room temperature. And we can directly mix with the cells. And you don't have to use the cell culture medium. You can mix with the PBS. And after that, we just withdraw these cells into a syringe and let them stable at room temperature for like 5-10 minutes. And as you can see that after 5-10 minutes, it formed the gel. However, because of this unique shear thinning property, the gel can flow like liquid during the injection. And right after the injection, if we flip this tube over, you will see that the gel is recovered, become a gel again. So you can imagine that with these properties, that you can prepare the gel at room temperature with peace in mind that the gel can easily to homogeneously mix with the cell. And then the hydrogel is starting to cross-link. The cell can be homogeneously to suspending inside of hydrogel. And because that we are able to stable the gel, and the gel has the shear thinning property, you won't crop the needle. So you can prepare the sample in bulk, and then you can do the injection slowly. You won't crop the needle. So this unique property can also retain a very high cell retention after the injection. We also have biocompatible data showing that the gel has no toxicity. The gels that pass of these pathogens, the testings with negative result, is also biocompatible with no side of the toxicity. And also after 8 weeks of the in vivo incubation, there's no inflammatory response. So gel itself is very, very safe for the injection application. Most of people that when they're using the hydrogels, traditionally they will use like some animal-based hydrogel like VitroGels to do this experiment. And to compare with these two systems, one thing is that this is the xeno-free system, but we modify it with the biological function. And it has the room temperature stable, so operation-wise, it's much easier. We are able to manipulate our hydrogel systems, which means that we are able to control the mechanical strength. If you need that mechanical strength stronger, we are able to manipulate this hydrogel. We can also manipulate the functional ligand degradability of the hydrogel. So it's the full tunable systems. instead of the animal-based systems that even some people say that we use these high concentration one. It only means that the protein concentration is higher. That doesn't mean that the whole mechanical properties is stronger. And because of this pure system, we are able to control these growth factor supplement compound. So you don't need to worry about these 2,000 different compounds in the animal system. You are able to add whatever you think is suitable for these injections to the mixtures. For the cell harvesting, the downstreams, we can do that easily, batch-to-batch consistency, and easy to adapt the lab automations for this in vitro application actually. And our product is always in stock. So that is some significant applications that when people are considering doing these injections with synthetic hydrogel. And on another critical points then these days, when people start to use the hydrogel system to do the PDX or PDXOs, the patient-derived organoids culture system, considering that when we get the tissue from the patients and putting into a pure system, well-defined system, will reduce the risk to changing the phenotype of the sample. And especially that if we can use the same system to do the animal injection, and after the injection, we can take the same sample out and embedding into the same hydrogel system for the organoids culture, then that is the big advantage to make the consistency of the sample and the testing result. So with that, I think that we've also mentioned about the few future properties, the comparisons. Cold temperature, we don't need it. But if you prepare the samples and want to put the whole mixings in the ice bucket, you can do that too. The gel won't clog the needle, so even you adjust the temperatures higher or lower, the gel is still injectable. And xeno-free systems, and so because of this injectable property, we can do that long-term injection, and smooth of the injection won't be able to worry about the needle clogging issue, and full control of the ECM system. All right, so we have some data that with collaboration with some organization and showing that the hydrogel can directly compare with the VitroGel for a CDX model. Here is some example of the lungs tumors and prostate cancer, and also that we also have this PDX model with like lungs tissue fragment and embedding in the hydrogel. And as you can see here, after the weeks of growth, the tumor growth is faster in our hydrogel system, and also the final size of the whole tumor is bigger. So with that, the system is excellent for the drug discovery, drugs testing study. As you can see, that some publication here showing that this hydrogel can be easy to inject and observe under the imaging system, and also that after injections, that we are able to see the tumors growth and in comparison with the different drug treatments. This is another paper has been recently published at Journal of the Biomedical Science. This is the ectopic tumor models. So instead of doing the subcutaneous injections, these groups they do these ectopic injection by mixing the hydrogel and the pancreas cells with a 29 G's needle. They are able to inject these hydrogels into the mice, the tail of the pancreas with the 1 million cells injection. And as you can see here, the tumor grows nicely. They can also use these models the same to compare with the drug treatments as well. And as we describe a lot of these in vivo models, our hydrogel system can also support the in vitro models. And these will give a very good connection between the in vitro data and the in vivo data. And here we give some example of these popular cell lines used for the Xenograft application. And with the same cell lines that putting in the same hydrogels in the in vitro application, we can see the, for example, here is the spirals, the growth and the invasion assays formed in our hydrogel system. We can also grow like these colon cancer cells in our hydrogel, make it into the 3D structures, same as this the pancreas, the cancer cells model, and monitors the cell growth in vitro. So this is a system that we are able to smoothly connect the in vitro data and the in vivo data in the same hydrogel system. And the table here that shows that some of these successful and most popular used cell types with our hydrogel system. And as you can see here, we have multiple different hydrogel can support this application. But here is the one of the most popular hydrogels. We call it the VitroGel Hydrogel Matrix. The SKU number is VHM01. And this is one of the hydrogels that have, is ready to use. So people just need to prepare their cell suspension and mix with this hydrogel. They don't need to adjust the gel concentration, adjust the other factor. So it's ready to use, very simple, and can support the very nice these tumor growth. And also that, as you can see here, this is one of the stroma cells growth in this hydrogel system and can directly compare with the animal-based hydrogel. And you can see that this stroma cell can bind into the hydrogel matrix and form the very nice 3D structures. So from the in vitro to the in vivos, these hydrogels can support a wide range of the cell types. And beyond that, we also have over 250 different cell types can support by our hydrogel systems. With that, I would like to give the rest of this webinar to Minchi, and he will tell us more data from HD Bioscience. Thank you.
Okay, hello. My name is Minchi, and I'm a scientist from HD Bioscience, a Wuxi Apptec company, and we are the in vivo pharmacology department. And I'm glad to share our data with the TheWell Bios. We established the VitroGel system with animal CDX model cell-derived xenograft and compared to the animal-based ECM. Can we go to the next slides? Yeah, so here it's we started collaboration to fund a series of studies, establish the human cancer CDX model using either VitroGel and compare with the traditional animal-based ECM. And the goal is to first establish the VitroGel system CDX models in mouse, and we pick different types of human cancer and all compare with the animal-based ECM. So for those models, we all use asemic nude mice, and the cell suspension was mixed in PBS 1 versus 1 mixed with VitroGel or hydrogel and implant subcutaneously in the mouse right lower flank. Can we go to the next slides? Yes. So here is showing the tumor growth curve with 3 cell line. HCT116 is the colon cancer, H8006 is breast cancer, and the CALO6 is lung cancer. Those 3 are very fast growing models. And as you can see in those models, the control animal-based ECM and versus the G2 VitroGel-based VitroGel groups, the xenograft growth curve is similar. There's no significant difference. Indicates the VitroGel system works perfectly with those tumor lines. But however, you may ask, those fast-growing tumor that are aggressive, even without VitroGel or any ECM, they can grow as fast as they are. So, and please go to the next slides. We also test some slow-growing models, like PC3 is a prostate cancer. Like most prostate cancer CDX models are very slow, and the HeLa cell is the Calvis hole cancer, and the third one is the PANC1 is the pancreas cancer. Those are very slow models, as you can see, especially HeLa and the PANC1. They took 16 days to reach the size of 300 to 400. But still, with those slow-growing model, the VitroGel group shows the similar growth curve in compare with the animal-based ECM. And more advanced, so the TheWell Bio is amazing. They can design specific modified VitroGel, depends on our study design and the special purpose. So let's go to the next slides. So this is an example of CCRF, CM. This is a leukemia human cancer cell line. So this model is tricky because leukemia is not it's suspension cell in culture. It's very hard to form the xenograft in mouse. So this kind of xenograft are very soft, and without ECM supplies, it takes very, very long, or even sometimes they fail to develop a xenograft. So in this case, we designed the modified special hydrogel, and we compare it with the traditional ECM. The growth curve, as you can see, are similar. So that indicates the VitroGel system works with the most common types of human cancer CDX model. And for special cell line, we can always design a modified hydrogel for special purpose. And some projects we are undergoing working with TheWell is we gonna further use a modified hydrogel to so some human cancer cell line that are very slow growing in mouse, or sometimes they don't grow in mouse. So we want to see if with modified hydrogel they can grow even better than the traditional animal-based ECM. Sometimes if the cell line doesn't work well with VitroGel, we may still have a modified hydrogel to make the cell grow fast in animal model. So that's something we are working with. So as a summary, can we go to the next slides? Yeah. So just a brief introduction of us. We are the HD Bioscience, located in San Diego, California, a Wuxi Apptec company. Our in vivo department provide over 100 CDX model surface, covers most of the human cancer type, and also we have the syngeneic model, orthotopic model, metastatic model for different goals of immuno-oncology research. And we also have other model like chemical-induced model for high-fat delta metabolic disease, NASH, fibrosis, and we have humanized model using NSG mice with the human PBMC engraftment. This can mimic the human immune system in mouse and further test the immunotherapies. So that's everything I'd like to share with you, and can go to the next slides. Yeah, I think maybe John, you can give a summary. Yeah, thank you, everyone.
Thank you, Minchi. Yeah, so we do have a web page talk about this Xenograft application. So if you're interested to learn more about these technology and see that the protocol, and we also have the video protocol to show you how to use it step by step, please visit our website. And we also list a whole bunch of the FAQs questions that people may have by using the xeno-free hydrogel system to replace the animal-based ECMs for this application. We also have these success sale table, success case study, and publication on this webpage. So if you need more information, please visit our website. We also have several different application notes talk about on this topic, including the CDX model, PDX model, and some of these publications as well. So in summary, is that this is a synthetic hydrogel system, very easy to use, can replace and close mimic the real ECM system, and supports the multiple different cell types to growth. It's very easy to use, and it can support not just for the xenograft application, it can also support these tissue engineering for those people that want to use the stem cells, mesenchymal stem cells, or even use this system as a injector system for control release, is all doable. And the most important thing is that the synthetic system we have, we can go for the GMP. We can go for a clinical application. So with the GMP system. So if you want to open the door for the application for downstreams, for the clinical application, and this synthetic functional hydrogel system is the way to go. With that, I would like to thank you, and we're happy to take the questions. Thank you so much.
Thank you, John. I think there are a few basic questions that question and answers are, do I have to work on ice with your system? And I think you might touch base on that, but go ahead.
Yeah, you don't have to. The hydrogel is room temperature stable, so you can typically when we do that, you can bring the gels from the refrigerator to the room temperature, and you can mix everything at room temperature. But if you think that you want to use the ice bucket, I know that some people come to us and say we still want to keep the gel in the ice bucket. It's doable. After you're mixing with the cell, you can still keep everything in the ice bucket and then do the injection. So that shear thinning properties will maintain. So you won't you won't change because that you put the gel in the ice bucket or you keep it at room temperature. The shear thinning property is still there. So you still make the hydrogel is very easy to inject.
Okay, thank you. I have another question from attendee. Have you tried the VitroGel on IPSCs cells?
Yes, we even have a hydrogel system called VitroGel Stem, which is our optimized hydrogel formulation for IPSC cells. So we do have the hydrogel can support the IPSC growth, and we use that hydrogel for IPSC 3D cell culture or IPSC 3D expansion. And we can also directly differentiate iPSC in that hydrogel system by adding different molecules to induce the differentiation. And we even have one workflow to show the people how to use that IPS hydrogel system to differentiate the cells, and then embedding into our organoid hydrogel to make the organoid mature. And also, all our hydrogel system, we have two big group of hydrogel system. We have a group of hydrogel we call ready-to-use hydrogel, which that we optimize the gel formulations for different applications and different cell types. And we also have another group of hydrogel, what we call the high concentration hydrogel, which is designed based on the gel properties, like mechanical properties, like functional ligands or degradability. And those hydrogels, you can use it like a Lego block. You can pick a different formulation and mix and match together. But no matter is that ready-to-use hydrogel or a high-concentration hydrogel, it's all injectable. So which means that all our hydrogel system can do this injection easily.
Thank you, John. I have another question from the attendee. When mixing cells with the VitroGel, does the gel generate bubbles?
No, actually, the gel is when you first take the gel, it's like in a liquid form, and then so when we mix with the cells, it will increase the viscosity, but the viscosity increasing won't be like dramatically increasing, so which will maintain a lot of little bubble in the hydrogel. So the increase of these viscosity when we mix with the gel with the cells together, the cross-links is slowly formed. So it will gently form the hydrogel without the air bubble forming. Even like when we use like some high concentration hydrogels, sometimes you might see the air bubbles that you introduce, but these air bubbles can easily either like you keep it as a warm temperature, 37, you release this air bubble, or you can simply just centrifuge the mixtures to get rid of the air bubble. But in most of cases, in most of our ready-to-use hydrogel, if you use it at room temperature, you won't get the air bubble.
Okay, good. How long can the VitroGel cell mixture retain its injectable status when mixed and comparing it to animal-based, and how will that help in the workflow?
Actually, as long as your cell can survive, we have so we have testing these like when we mix the hydrogel and the medium without the cells, it can maintain these injectable states for months. So which means that as long as your cell can survive in this environment, the injectable status of the gel can maintain. So we can maintain this injectable status for like hours if you want to do like a very long-term injection. That's no problem. And we do this test, we just mix the gel and the medium together or like PBS together, and that hydrogel status can maintain for months, no problem.
Sujata asked another attendee, just following up on the same question on the IPSC question. Just to follow up on the last question about IPSCs, you mentioned you grow them in 3D cultures. So does that mean that they are growing in spheroids?
The iPS cells, we culture them, they can form like 3D spheroid structures in our hydrogel. We also can induce these cells to growth and differentiation in the IPS hydrogel VitroGel stem in that system. But that is just because that the IPS, the colonies we sitting in the hydrogel, they prefer to forming as a spheroid structure. That doesn't mean that all the other type of cells sitting in our hydrogel system will become like a spheroid, because it really depends on the cell's properties. For example, if we sit in like fibroblast cells in our hydrogel system, and because the fibroblast cells, they like to interact with the matrix, so it will form in like a fibroblast structure will form the cell networking structure instead of the sphere. So the morphology of the cells growth in our hydrogel is really depends on the cell types and the cell's properties.
Okay, I have a two-part question. I think probably three from Virat. Is the matrices toxic? Is it manipulated biodegradable?
No, the toxicity testings, the ones that I think that we have one part of data showing that toxicity testing go up to like 8 weeks, and actually we have 6 months data and showing that this hydrogel is very stable and it's very safe. There's no like no signs of these toxicities when we inject to the animal. Biodegradable, we are able to manipulate the hydrogel. So one part of the manipulations is that we have a hydrogel system we modify with the MMP sensitive ligand, and so that hydrogels can degrade much faster than the traditional than the regular hydrogel. So with that, we are able to manipulate the degradability of the hydrogel by using the different ratios of the mixings of this MMP hydrogel, or like using 100% pure of the MMP hydrogel. So the gel is degradable in animal, and we are able to manipulate the ratio of the degradation.
And I think that ties into one of the questions by Laura. When injecting in vivo, is the gel degraded over time? And if so, how soon after injection would you expect no gel to be left?
So yeah, we see the data when we manipulate hydrogel with the high concentration of these MMP ligands, that we will see that the gel can degrade, and also it also depends on the concentration of the gel. So if you want to use the high concentration, of course it will degrade slower. If we do like a lower concentration degradation, we'll see that 50% of the gel will disappear within like two weeks to 3 weeks. That is one type of the experiments we can do. We have done, and we are able to manipulate that part as well. And on the other hand, the degradation of the hydrogels inside of animals is also depends on the cell types you mix with the hydrogel. So if the cells has the secretes the more enzymes and degrades the gel, the degradation might be faster.
Okay. Can TheWell Bioscience, as probably another can do GMP?
We can provide the GMP grade hydrogel system. Currently, we take like a customer order and say they want the whole bunch of the materials in GMP. We take like kind of the customer orders in GMP grade.
Okay. I believe it was spoken, but someone asked is, can TheWell Bioscience custom modify hydrogels?
Yes, as I described, we have a hydrogel system that we call the high concentration hydrogel that is modified based on the hydrogel properties from the mechanical strengths to like the different functional ligand, degradabilities, all these we can change. So we are able to customize the hydrogel. We even have the customers, they know that some particular functional ligands they want, and it's very important for their application. So we are able to binding that ligand to our hydrogel system, maintain the whole system has these very easy to use injectable property, but include these important functional ligand for them. So we are able to do the customized hydrogel.
Great. And I think one of the basic questions is, do you have stock issues?
No, we don't have this stock issue. So our gel is pure synthetic. So even we need to like start everything from the scratch, the lead time is about like 2 to 3 weeks. We can have the everything to the market. So we don't have this stock issue.
Okay. I do not see any other questions. If you guys have any other questions we want to answer, please chat it in. And if not, that we will conclude this webinar. And if you have any questions about this webinar, please, the contact information is here. You can call us or email support. And also HC Bioscience, their contact information is there as well. And we will also have this webinar recorded, so you will have this email to you at the follow-up. And we will also follow up with any questions that you may have with us. So thank you, everyone, for attending this webinar. We hope you find this helpful, and we will have future webinars. Please follow us and subscribe to our newsletter for future webinars. Thank you so much.
Thank you, everyone.
Thank you, everyone.